Previous session, we discussed in general the components of cartilage. To recap, we discussed introduction, components like cells which are chondroblasts and chondrocytes, extracellular matrix which included fibers and ground substance, and finally the arrangements of these components in general. In this session, I will be explaining about each type of cartilage in specific. Now coming to the types of cartilage, depending upon the presence or absence of perichondrium, number and arrangement of cells, varying proportion of fibers and varying amount of ground substance, it is divided into three types: hyaline cartilage, elastic cartilage and white fibrocartilage. Hyaline cartilage, it is the most abundant type of cartilage in the body, appears whitish blue and semi-transparent in gross appearance. It gets its name because it is transparent. Hyalose means glossy. Hyaline cartilage might calcify during old age, the most being affected are costal and large laryngeal cartilages. Articular cartilage, a type of hyaline cartilage without perichondrium, remains as cartilage throughout life. When we look at the structure, hyaline cartilage is covered completely by perichondrium. Perichondrium as outer fibrous layer formed by collagen type 1 fibers along with fibroblasts, and inner chondrogenic layer with fibroblasts and chondroblasts. Perichondrium is absent in articular cartilage. Articular cartilage is similar to the rest of hyaline cartilage without perichondrium. Coming to the matrix is homogeneous and basophilic. It has type 2 collagen fibers. The collagen fibers cannot be distinguished from the matrix and looks homogeneous in appearance because the refractive indices of collagen and the ground substance are same. The ground substance resists compressional forces and the collagen fibers resists tensional forces. The collagen fibers are arranged in a manner it resists tensional forces. The cells are arranged singly in the peripheral part, which are ovoid in shape, and the long axis of the oval cell is parallel to the surface of the cartilage. In the inner part towards the center, the cells are arranged in clusters or groups. These groups are called cell nests or isogenous groups. Each cell nest contains 2 to 8 mature chondrocytes. All the cells are present in the space called lacunae. Around the lacunae, the matrix is more intensely basophilic and is called territorial matrix or lacunar matrix. In between the territorial matrix, the matrix is lightly stained, less basophilic and is called interterritorial matrix. How are the cell nests or isogenous group formed? Or how is cartilage developed? During development, the chondroblasts start producing the extracellular matrix around it. As the matrix starts increasing in amount, the cells start separating from each other and get arrested between the matrix. As this cell starts dividing, it multiplies into 2, 4 or 8 cells. Since cells are arrested in the matrix, it cannot move away from each other and form a group of mature chondrocytes. Because of its smooth lubricated surface and resiliency, cartilage provides shock absorbing and sliding regions within the joints and facilitates the bone movements. Growth in bones occurs in two ways, either longitudinally, which increases the length, or increase in width of bone size. When chondrocytes secrete new matrix, Within the cartilage, it causes the bone to grow in length. This increase in size of length of bone is referred as interstitial growth. When chondroblasts along existing surfaces secrete new matrix, it causes the cartilage to expand and widen. The increase in the pre-existing diameter or size of the width of the bones is referred as appositional growth. This is the photograph of the section of hyaline cartilage to show how it appears under the low power of microscope. The two layers of perichondrium, outer fibrous and inner chondrogenic layers, are observed surrounding the cartilage. The matrix is basophilic and homogeneous in appearance. Just deep to inner chondrogenic layer, oval-shaped single chondroblasts can be seen oriented parallel to surface of cartilage. Deep towards the center of cartilage, isogenous groups of cells, the cell nests, can be observed. This is the photograph to show how the section of hyaline cartilage looks under high power of the microscope. It is focused towards the center of cartilage to show cell nests, territorial and interterritorial matrix. The cell nests can be clearly observed. 2 cells, 4 cells can be seen which are arrested in the surrounding matrix inside the pale stained lacuna. Okay, these are the spaces, okay, in which the chondrocytes are lodged. Now, these lacunae is very clear because of the shrinkage of the cells. In the living, the spaces are completely occupied by the chondrocytes. Surrounding the lacunae, the matrix is intensely stained, which forms the territorial matrix. In between the cell nests, the matrix is lightly stained and is called interterritorial matrix. Hyaline cartilage is located in developing skeleton as cartilage plates. In adults, it is present in articular regions in the form of articular cartilage lining the bones forming the joints. Then it is seen in costal cartilage, nasal cartilages, thyroid, cricoid, arytenoid. All three belongs to larynx, then trachea and bronchi. Please note the parts mentioned are related to growth, joints and respiratory system respectively. Okay, coming to the elastic cartilage. It is present in places where more flexible support is needed. It has flexibility than the hyaline cartilage. It appears yellow on gross appearance due to the presence of elastic fibers. Now, coming to the structure of elastic cartilage, it is similar to hyaline cartilage with some differences. Just like hyaline cartilage, the whole cartilage is surrounded by perichondrium with outer fibrous and inner chondrogenic layers. Instead of homogeneous matrix as in the hyaline cartilage, the elastic cartilage has numerous elastic fibers in addition to collagen fibers in the matrix. Cell nesting is not as much as in hyaline cartilage. Chondroblast, as in hyaline cartilage, is oval in shape, present at the periphery in single. Towards the center, chondrocytes are present in single cells or 2 cells in a group, lodged in spaces called lacunae. Chondrocytes are closely packed as the number of chondrocytes is more and amount of matrix is less. This is the photograph of the section of the elastic cartilage under low power of the microscope. The two layers of perichondrium, outer fibrous layer and inner chondrogenic layer, are observed on both the sides of the cartilage. The matrix is rough with abundant elastic fibers. Deep to the inner layer of perichondrium is the chondroblasts, observed singly, and towards the center are chondrocytes, present in two cells as isogenous groups or cell nest, which are lodged in the lacunae. Numerous cells with less matrix can be seen in this section. This is a photograph of the section of the elastic cartilage as seen under the high power of microscope. It is focused to show elastic fibers and cells. Elastic fibers are clearly observed in the matrix, and chondrocytes in groups of two are observed as cell nests. Elastic cartilage is located in external ear, auditory tube or eustachian tube extending from ear to nasopharynx, epiglottis, corniculate, and cuneiform. All three belong to larynx. All these are either related to production or reception of sound. Coming to the third type of cartilage, white fibrocartilage or just fibrocartilage, it provides tensile strength in the region where it is located. Coming to the structure, it is devoid of perichondrium, which is replaced by the connective tissue sheath. It consists of numerous rows of collagen fiber bundles and minimal ground substance. Collagen fibers present are type 1 and are oriented in the direction of the stress. Chondrocytes are few and arranged in rows between the layers of collagen within the lacunae in the matrix. This is the photograph of the section of white fibrocartilage as seen under low power of microscope. The section shows the chondrocytes present in linear rows between the collagen fiber bundles. The ground substance is very minimal surrounding the chondrocytes, which may be seen or may not be seen in the section. This is the photograph of the section of white fibrocartilage under high power of the microscope. The section is focused to show the enlarged view of chondrocytes in linear row surrounded by the collagen fiber bundles and minimal ground substance around the chondrocytes. White fibrocartilage is located in intervertebral discs, pubic symphysis, which are present in the midline of the body. The other locations are articular disc of temporomandibular joint, menisci of knee joint, glenoidal labrum of shoulder joint, and acetabular labrum of hip joint. With this, we have completed the specific features of hyaline, elastic, and white fibrocartilage. In the next session, I will be discussing on clinical anatomy of cartilage.